Hello everyone. Today I'm going to talk about assignment number 8. It's a kind of practical assignment which can be useful for you. We can also ask questions about assignment 8 in the final exam because you will have internet access during the exam. So please pay attention to this assignment as well. So if you want to use protein data bank, you should use this URL, and you can simply search your protein here in this box and click on search, and you can look up for the protein that you are looking for. For example, in this question we asked you for this specific protein, so simply type it here and look up for it. On the left side, you can see the 3D structure of this protein. So you can simply save this picture and copy paste it on the file to show us, for example, what's the 3D structure of this protein. And there are other information on this page which are very useful, and we will talk about them one by one. If we ask you about the primary structure, it means that we are asking you about the sequence of amino acids. So in that scenario, you should click on structure here under the picture, and the page which will be open for you will be the primary structure of the protein. So you can see the sequence of amino acids here. And if you click on each amino acid, it even shows you on the picture that where is the amino acid located. And this is very interesting. So you can just go through it and just play with it. Click on any amino acid to see, for example, where on the picture it shows. And it gives you much better idea about the difference between primary structure and 3D structure. And why this is important? Because, for example, when you look at this one and this one, these two amino acids at the beginning, you would say, okay, V and L are close to each other. And for example, P is far away from them. But maybe when you look at the 3D structure, you understand actually V is closer to F, for example. So these things are very interesting, and it also shows the atoms. In molecular level, everything is clear there. And this is also useful to know that everything that we are talking about is based on them. They are not letters. As computer scientists, we are showing them like letters like this, or as bioinformatician. But we should know that this is not the case in nature. Okay, and if we ask what's the length of its primary structure, even from the sequence, it's obvious. Based on these numbers, you can understand the position of these amino acids. For example, 21, it shows for 2 letters if you look at it like this. So the first letter actually is 21st. We should be careful about it, because when the number has more digits like this, we think maybe it's related to 3 letters. Okay, which one should I choose? It means that the first digit that you can see here is related to the position of that. Okay, for example, this amino acid is the position 111. This amino acid is 121 position. So I understand it from the first digit. These things we should be careful and notice from the beginning, because it's easy, as you can see, but sometimes it can get confusing if we didn't think about it before. So if we ask what's the length of it, okay, this letter corresponds to this position, so this is the length of this primary structure. Also, there are other parts of the page that you can find the length. It's written there. I will show you. Now, how many lysine amino acid can be found in 6K9 primary structure, and at which positions? So we should know for each amino acid with which letter we are writing them. Like for example, which letters corresponds to lysine, which letter corresponds to another amino acid. We should know about them. I also provided for you in one of the slides. I don't know whether it was in lecture 1 or lecture 2, but it's already provided for you. I also mentioned sometimes they show it with 3 letters together, but in sequences we use the 1-letter format. So for lysine we show it with K, for example. So we should check how many K I have inside my sequence. So simply you should count them. For example, one is here, the other one is here. You count them and you say, for example, the frequency of lysine is this much. Then you report the number of Ks which happened here. And at which positions? So you can also mention at which positions you observe them. For example, for this one, 1, 2, 3, 4, 5, 6, 7. At 7th position. Again, here I have 11th. And you write the rest of them. You find just simply Ks and report their position. For example, we have another one here, and you continue like that. It's pretty easy, right? Just specify exactly what's the number of them and what are the positions. Exactly right. For example, the positions of lysine is, then 7th, 11th, and so on. All right, in which organism this protein can be found? If you go back to the first page when you search this protein, then you will see here it's written Homo sapiens. Homo sapiens means that we are talking about organisms of human beings. So this protein is actually alpha globin subunits of adult human being. If we show it like this, it has 2 subunits of alpha, 2 subunits of beta in adult human being. And we are talking about these subunits. And even this one, they are the same. So, well, as a computer scientists, usually when we hear about such proteins, we don't know what they are, right? We don't have any idea. So this data bank is very useful for us, because if we are doing a research and we understand, we hear a protein for the first time, then we can get idea by this. And also biologists can claim that they know every details related to each proteins, because it's a vast number of data. And that's the reason they need computer scientists, because now during last 40 years especially, they have gathered lots of data, and we will talk about micro data analysis very soon, microarray analysis, and we will see that why they need computer scientists and mathematicians more than ever, because simply the number of data is vast, and it's very hard to handle everything and analyze the data. Okay, so when we ask in which organism, simply you say Homo sapiens, and that's the answer. And if we ask you which genes are known to be responsible for this protein production, then here it is written. If you go through the page, the same page actually, you just scroll down and you reach this part. Inside this subsection, you will see that it's written about some details which are important. Here it mentioned about the molecule, and the chain contains such subunits of alpha inside the hemoglobin. And here is also a tip for you, that it says that the organism is Homo sapien, and here you can see the responsible genes for that. HPA1 and HPA2 are responsible for production of this protein. So from here we understand two different genes are responsible for synthesizing alpha protein. It's not always the same case. Sometimes we see only one gene is responsible for the whole protein, like for example for beta globin gene, HBB, but also biologists cannot say 100% this is the case. Like this gene for 100%, I'm sure everything inside this protein comes from this gene. But until now, that's what is assumed. And based on the codons that we talked about before, you can see the trend in amino acids and the combination of amino acids synthesizing that protein. So now biologists are confident to say, okay, this gene is responsible for that. But in the future, if you hear, for example, there is another gene which is responsible for synthesizing that protein, don't be surprised, because it keep happening. Fortunately for hemoglobin, it's well studied, and it's pretty simple comparing to other proteins. That's why they are more confident. For example, for this one, we didn't see that much change in regards of hemoglobin during the last years, for example. And here also you can see the length of the sequence in the primary structure. Even from this table, you could answer the previous questions as well, right? If you click on this part, it will show you the secondary structure. I won't ask questions about that in the final exam, but please go through that too. Just take a look, because it's very interesting. We talked about secondary structure, and we divided it into 3 parts. And if you click on it, you will see that how we can categorize such sequences as well. And when we talk about mutations, here, for example, mutation is considered to be 0. This is the control case which synthesizes this protein. If we have a specific mutations, particular mutations on our gene, mutations, by the way, don't forget that we are talking about the gene level. There are other types of mutations also we mentioned, like on even chromosome. The whole chromosome sometimes is missing. So, but here I'm talking about gene level. Like we have sequence of nucleotides like G, C, C, A, and so on. And this, for example, nucleotide is mutated. Let's say instead of A, now we have another C. And because of that, this codon maybe synthesizes new amino acids, and the whole structure of the protein change, and we will have a different protein. We don't have this one anymore. So if we name them and they say, okay, this is, for example, 2 mutations here or 1 mutation here, then they will write it here. That's what it means in this table. Now, with which method the 3D structure was estimated? Here you can see for the experimental data snapshot that it is mentioned here. They used X-ray in order to come up with this 3D structure. And let me also show you this part. I won't ask questions about it in the final exam about this part, but you should know that this is related to statistical significance. And they mention here how accurate is the 3D structure that you can see here. And if you notice, these numbers shows that nothing is 100%, we could say. Even if something is mentioned about 100%, it means that, for example, we are talking about a very small p-value like this, for example. So nothing is 100%, and we are trying to gather data as much as possible with different methods. Here, for example, this is the method used here. This is not the only method to get the information about the 3D structure. It's not easy to get an image of a protein. They are gathering information to come up with this picture. So we should have it in our mind that nothing is 100%, and we are talking about probabilities, about p-values, and we talked about it briefly before. Okay, guys, if you have any questions, please let me know. Otherwise, we will talk about microarrays analysis in the next session. Yes, please.
For this structure, we had two structures, the alpha structure and the beta structure.